Welcome to G Fun Facts Online. Today, we're diving deep into the incredible story of a medication that revolutionized cancer treatment, Taxol. This isn't just a story about a drug. It's a 30-year saga packed with scientific breakthroughs, daunting ecological challenges, and the sheer power of human ingenuity. It's a journey from a chance discovery in a forest to the cutting edge of biotechnology, all in the fight against cancer, and all while learning how to protect our planet. So, how did a tree bark change the world of medicine? Our story begins way back in 1962. Imagine a botanist, Arthur Barkley, working for the U.S. Department of Agriculture. He's out in Washington State's Gifford-Pinchot National Forest, collecting samples as part of a massive National Cancer Institute program. They were looking for anything in nature, plants, animals, that might have anti-cancer properties. One of the samples Barkley collected was from the Pacific yew tree, Taxus brevifolia. 2 years later, in 1964, researchers Drs. Monroe Wall and Mansukwani over at the Research Triangle Institute in North Carolina found something intriguing. Extracts from the Pacific yew's bark were toxic to living cells. This was the first hint. It took several more years of painstaking work, but by 1971, Wall and Wani had isolated the active compound. They figured out its complex chemical structure, C47H51NO14, for yew chemistry buffs, and named it Taxol, derived from the tree's genus, Taxus, and because they thought it was an alcohol. But just knowing a compound is toxic to cells isn't enough. How did it work? The real breakthrough in understanding Taxol's power came in 1979, thanks to Dr. Susan Band Horwitz, a molecular pharmacologist at Albert Einstein College of Medicine. She discovered Taxol's unique mechanism of action. At the time, other drugs that targeted cell division, known as antimitotic drugs, usually worked by preventing tiny structures called microtubules from forming. Microtubules are like the cell's internal scaffolding, crucial for cells to divide. Taxol did the opposite, and this was revolutionary. It stabilized these microtubules, essentially locking them in place and preventing them from disassembling. Think of it like this: if cell division is a carefully choreographed dance, Taxol freezes the dancers mid-move. This effectively halts cell division, especially in rapidly dividing cancer cells, leading to their death. This novel approach made Taxol an incredibly promising candidate for cancer therapy. As Taxol moved into clinical trials in the 1980s, its potential became even clearer. It showed remarkable activity against tough-to-treat ovarian cancer and metastatic breast cancer. But a massive problem was looming: supply. The Pacific yew is a slow-growing tree. We're talking trees that are often 200 years old. And the main source of Taxol? It's bark. Harvesting the bark kills the tree. Estimates suggested that you'd need the bark of 3 to 6 mature yew trees just to treat a single patient. Imagine the scale. If Taxol was to become widely available, hundreds of thousands of these ancient trees would have to be sacrificed. This posed a severe threat to the Pacific yew species and its entire ecosystem. The environmental cost was simply too high. The National Cancer Institute was facing a critical supply crisis. They had a miracle drug, but couldn't sustainably produce it. This ecological dilemma sparked a global race. Scientists everywhere scrambled to find alternative ways to get Taxol without destroying forests. This is where the ingenuity really shines. A major breakthrough came with semisynthesis. French chemist Pierre Potier and his team discovered that a precursor to Taxol, a compound called 10-deacetylbaccatin-3, or 10-DAB for short, could be found in relatively larger quantities in the needles and twigs of the European yew, Taxus baccata. This was fantastic news because the European yew is more abundant, and you don't have to kill the tree to get its needles and twigs, a much more renewable resource. This 10-DAB could then be chemically converted into Taxol. Around the same time, Dr. Robert Holton at Florida State University also developed an efficient way to make this conversion. In 1991, Bristol Myers Squibb, the company partnering with the NCI for Taxol's commercial production, licensed Holton's technology. This semi-synthetic approach became the dominant way to produce Taxol, taking a huge amount of pressure off the wild Pacific yew. However, it wasn't perfect. It still needed large yew plantations, and used a lot of toxic solvents in the process. Now, for the hardcore chemists out there, the ultimate challenge is total synthesis, making a complex molecule like Taxol from scratch using simple lab chemicals. Taxol's structure is incredibly complex, a real beast for chemists. Despite this, in 1994, two teams, one led by Robert Holton again, and another by K.C. Nicolau, independently announced they'd done it, the first total synthesis of Taxol. This was a monumental achievement in organic chemistry, a true testament to what chemists can do. But these processes involved many steps, and were far too complicated and expensive for making Taxol on a large industrial scale. A much greener and more sustainable path forward emerged with something called plant cell fermentation, or PCF technology. This is super cool. Instead of harvesting trees, scientists cultivate taxus cells in large bioreactors, like giant high-tech vats, under controlled conditions. These cells naturally produce Taxol and its precursors. Phyton Biotech was a pioneer in this area. Bristol Myers Squibb also adopted this technology. PCF has a lot of advantages: a consistent and reliable supply, a cleaner product, no need to chop down trees, and it drastically reduces the use of solvents and hazardous waste compared to semi-synthesis. Nature, it turns out, had more secrets. Scientists discovered endophytic fungi. These are microorganisms that live inside plants, in a symbiotic relationship. Amazingly, some fungi found living within taxus trees, and even some in non-asterisk taxus species, can also produce Taxol. Imagine tiny fungal factories making this life-saving drug. While the amounts produced by these fungi are often low, and the process can be a bit unstable, research is actively ongoing. Scientists are trying to optimize this using techniques like growing different fungal species together, or even genetic engineering. Figuring out how these fungi make Taxol, they might have borrowed the genetic instructions from the tree itself through something called horizontal gene transfer, is key to using them for industrial production. And that brings us to the very cutting edge: synthetic biology and metabolic engineering. Scientists are working to map out the entire biochemical recipe, the biosynthetic pathway that the yew tree uses to make Taxol. This pathway involves a whole cascade of enzymes and chemical reactions, many of which are still being uncovered. The goal is to identify all the genes and enzymes involved, and then reconstruct and optimize this pathway in more convenient host organisms like yeast. Yes, the same kind of yeast used to bake bread or brew beer, or even in fast-growing plants like tobacco. Imagine yeast or tobacco plants churning out Taxol or its precursors from simple, inexpensive starting materials like sugars. This approach promises to make Taxol production even more efficient, cost-effective, and environmentally friendly. We've already seen recent breakthroughs, with scientists identifying missing enzymes in the pathway and getting engineered hosts to accumulate Taxol intermediates. So, what's the upshot of all this? Taxol was approved by the FDA in 1992, initially for ovarian cancer that wasn't responding to other treatments, and later for breast cancer, lung cancer, and even an AIDS-related cancer called Kaposi sarcoma. It has become one of the most important and widely used anti-cancer drugs globally. It's even on the World Health Organization's model list of essential medicines. That's how crucial it is. The 30-year quest for an eco-friendly way to make Taxol has completely transformed how this vital drug is produced. We've gone from the destructive harvesting of rare ancient trees to sophisticated plant cell cultures and the futuristic possibilities of synthetic biology. This journey is a shining example of our continuous drive for innovation and sustainability in medicine. While there are still challenges in scaling up some of the newer methods for industrial production, the progress made means we have a much more secure and environmentally responsible supply of this life-saving medicine for generations to come. The ongoing research into how Taxol is made by nature and how we can replicate and improve that in the lab continues to unlock new possibilities in our fight against cancer. It truly shows the enduring power of combining nature's pharmacy with human scientific ingenuity. That's all for this episode of Fun Facts Online. Join us next time for more fascinating stories from the world of science.